Hello students, welcome to Distinction Learning Platform, and on this platform you find revisions, tests, expansion to difficult topics, a lot of stuff. So before we begin our lessons, make sure that you subscribe and like the video. Okay, to make the story short, let us begin. In this video, we're talking about leukocyte activation, and this is under what? Acute inflammation. If you haven't watched the video about acute inflammation, make sure that you watch it because we talked about it. And remember that, just to recap, acute inflammatory response is basically the protective response provided by what? By the body, because it was exposed by what? Foreign invaders, necrotic tissues, and after that, this inflammatory response is going to make sure that it eradicates necrotic tissue and as well as what? Foreign invaders. Lastly, it's going to initiate what we call repair, which is a very important concept. Okay. Now, what is the other important aspect that you need to understand? Okay, let's talk about now leukocyte activation. So leukocyte activation, it is under what? Cellular events, because in the other video we talked about the first part under cellular event, which takes part, which we call what? Leukocyte recruitment. Okay, make sure that also you watch that video. Okay, so now we'll proceed to what we call leukocyte activation. The question is, how do leukocytes activate themselves? Okay, how is that something like that is going to happen? Okay, if you look at here under cellular events of acute inflammatory response, those things we said to say a leukocyte will come here. So the thing that we talked about is how the leukocyte or the white blood cells comes out of the blood vessels and they go to the what? Interstitial space. Okay, so what are we saying? What we are saying is that the things that we talked about, if this is a neutrophil which is here, how does it move from the what? White from the blood vessels and go into the what? Interstitial space. We talked about that in the previous video. Now, what we're talking about, this is the interstitial. Already it is on the side of what? Is infection. It is on the what? Interstitial what? Space. That's why it is. Okay. Now, the moment it is here, because of those things that we talked about, the chemotaxis, then it is there where the site of infection is. As you can see, this is the foreign invader. Basically, let's just say that is a bacteria. Very important to take note of that. Okay. Now, the question before we dive into the details, okay, we need to understand what are the three important aspects under what? Leukocyte activation. What are the three important things that you need to understand? The first one is what? Phagocytosis. What is the first one? The first one is what? The only thing that's going to happen, phagocytosis. This is the first process that's going to happen, okay? Phagocytosis under leukocyte activation. What else apart from phagocytosis? The other aspect is what? Production of what? Nets. Production of what? Nets. Okay? Production of nets, which is what? Neutrophil extracellular what? Traps, which we talk about in what? Details. What is the last one? Production of what? Chemical mediators. So the last one is what? Production of chemical mediators. Okay? Production of chemical mediators, which we talk about at the end of the video. Okay. Now, since we've understood the three important aspect under what? Leukocyte activation. The first one is what? Phagocytosis. The second one is what? Production of nets. The other one is what? Production of chemical mediators, which we'll talk about in details. Okay. Chemical mediators that will come from plasma. Okay. The chemical mediators that will come from what? Cells. It's themselves. Okay. Now, let's start with phagocytosis. So the process of phagocytosis is a complex process, but we're going to simplify it so that you can understand fully. Okay? So what is the first thing? Like here we said, leukocyte activation contains these three important. If you know this, then that's basically under leukocyte activation. Now we need to know under phagocytosis, what are the three important aspects or cases that can happen under what? Phagocytosis. What is the first one? The first one is what? Engulfment. We'll talk about how the engulfment happens here. Okay? So what is the first one? The first one is what? Engulfment. Let's use the blue marker. The first one is what? Engulfment. Okay, the first one is what? Engulfment. That's going to happen. What is the second thing after engulfment? Okay. Then the other thing is what? Formation of what? Phagosomes. Okay. Formation of what? Formation of what? Phagolysosomes. So let's just say formation of what? Phagocytic what? Vacuole. So we said, so P phagocyte, so we say phagocytic what? Vacuoles. So we'll understand what does that mean. Okay, we'll talk about that. Then the last one is what? Now let's use this one. Let's use this one because this is interesting. So the other one is what? Killing now. Killing of the what? Any foreign invaders. That is the what? The last process that will happen. Okay. So those are the three important things that you need to understand about phagocytosis. Now let's begin with what? Engulfment. Engulfment is somehow complex, but we will simplify it, don't worry. Okay. So engulfment, what does that mean? Okay, this is let's just pick the famous one, which is what? Neutrophil. Although there are other leukocytes that you can do, macrophages, they can do phagocytosis. Okay, eosinophils, they can do basophils, they can do what? Phagocytosis. But let's just pick one, which is what? Let's imagine to say this is a neutrophil or a polynuclear type of what? White blood cell. So let's just say this is a what? A neutrophil. Okay, this is a neutrophil. Okay, if this is a neutrophil now, the question is, before engulfment happens, because to engulf is to get this foreign invader or a bacteria inside the cytoplasm of this neutrophil. That's what we call engulfment. Now the question is, how does it do? So before eating up that foreign invader, it has to what? Recognize it as what? Foreign invader. Now that's very, very important to understand about what? Receptor. These two time receptors. They are waiting to recognize this bacteria. So as you can see now, the question is, what are these receptors? What are the types of receptors that you need to understand? If you watch that video, we talked about the receptor that can be found on the leukocyte. Okay, we call them phagocytic what? Receptors. A good examples are what? Toll-like receptors, which are about what? 10. They are about what? 10 when it comes to what? The family there. And we also talked about the inflammasomes, chemical types of receptors. So there are a lot of receptors. So let's just call them phagocytic what? Receptor. Okay. So let's just call this what? Phagocyte receptors. So these time receptors. So these are what we call phagocyte receptors. These are the ones that are going to recognize this one. Good example what? Toll-like what? Receptors. Toll-like what? Receptors. Let's just put toll-like receptors for phagocyte receptors. Okay. These are the ones. Now, before they recognize like that, before they eat up that foreign invader, what are they going to do? So there is something that is interesting that they do. Okay, because let me give you a real life example. If you are eating something that is solid and it is big, okay, you are failing to swallow it. What are you going to do? You are going to drink water, then you put so that you can what? You can swallow that efficiently and without having any difficulties. Okay. Okay. So now, what do we call that after that example? So what do we call that? So what do we call substances that are going to make sure that this engulfment process is easier? What do we call that? We call them what? Opsinin. Okay. So what do we call those substances? So opsinin. So let us write them here. Okay. We call them what? Opsinin. So opsinin, basically, these are the substances that are going to make sure that this engulfment process goes smoothly. Okay, so that bacteria is not going to have what problem when it comes to what? Engulfment. Okay, opsinin. Now the question is, what are those good examples of opsinin that we have in our body? The first one is what? We know that one. What is the first one? We know the first one is what? The C3. The C3 what? Complement protein. The C3 complements protein, which can be found in the what? Blood plasma. Okay. What else? Also immunoglobulin like what? IgG. Okay. IgG also is one of the opsinin substance that is recognized. What else? Also another one, which is what we call leukolectin. Okay. collecting. Okay, these are the three important what opsonin substances that are going to what make sure that engulfment process is easier. Okay, now I talked about the receptors here. Okay, the toll-like receptors. Also, we've got so this is phagocyte, or this leukocyte or neutrophil. Let's say this is a neutrophil. This neutrophil, it also has receptors for C3, it has receptors for IgG, it has receptors for what? Collectin. What do we call that? So we are going to use here, okay, so let's write here, the C3, basically that is what? The complementary receptor type 1 and also complementary receptor type what? Type 3, okay, that is the receptor that can be found on the what? On the neutrophil membrane there. What else? Also for IgG also it has what? Receptors. We call them what? FC receptors. Okay, FC receptors for what? IgG. What else? Also for collectin, this one, the collectin is going to have a certain receptor which we call what? CQ1 type of receptor, also found on the what? Neutrophil. So those are the receptors for what? Opsonin. Okay, they'll come here. Okay, now let us talk about the engulfment process. Okay, this opsonin, we are saying they are going to make sure that this whole engulfment process, so this eating up of the bacteria, becomes easy. So what they do is like this. So let's just pick one. Okay, if this is an opsonin, it will come here and bind to this what? Bacteria. It will bind to this bacteria. Okay, and as well as it will go to the what? Receptor of that same opsonin. Okay, let's say this receptor. Okay, I'm not talking about the toll-like receptor. I'm talking about the receptor for this opsonin. Okay, what do we call this receptor? Let's say this is a C3 receptor or C1 receptor for a complement. Okay, and it's like this. So this is now, this whole process is what we call what? If the opsonin bind to the foreign invader like the bacteria, and it bind to its receptor on the what? Neutrophil or on the white blood cell like the C1 receptor here. Okay, what do we call this process? We call it what? Opsonization. Okay, this is what we call opsonization. Opsonization basically where the engulfment is happening in the presence of what? An opsonin. Okay, an opsonin is binding to its respective what? Receptor. Don't you get confused on that. Okay, if this happens now, this engulfment process is going to speed up, and what's going to happen now? There's something interesting that's going to happen. You've seen this neutrophil. This neutrophil is going to start doing something. To start what? Extending this cytoplasmic membrane, this one. To start extending like that, okay? To start extending this what? Cytoplasm like that. Also this cytoplasm like that, okay? Why extending that cytoplasm? So that now it can surround this what? Foreign invader, this bacteria. Let's just be calling bacteria. So it can surround this what? Bacteria, so that it can pull it into inside via the process called what? Like endocytosis. Okay, so what we are saying is like this. Okay, so let's make it clear. So what we are saying is like this. Okay, if this is a bacteria like that, okay, this is a bacteria, okay, it is like that. Then what's going to happen to this? It will start what? Extending its what? Cytoplasm around what? Around this bacteria like that. Okay. So this extension of this cytoplasm are what we call pseudopodia. Okay, these are what we call what? Pseudopodia. So these pseudopodia, these are just extension of the cytoplasm so that it can surround the what? This bacteria. Okay, if it surround now, it will complete surround the what? The bacteria like that. Thereby, if it's complete surround the bacteria, now it will enter into the what? This cytoplasm. That is what we call engulfment process. Basically, what I mean is like this. Okay, that is a bacteria. Let's say this is a bacteria. Then I'm a neutrophil. Okay, I'm a neutrophil. So this is a bacteria. Then what's going to happen is like this. I'm going to extend this pseudopodia, also another pseudopodia, so that I can complete cover this bacteria and pull it inside like that. Okay, yeah. So that's what I mean. So this is what is happening here. Okay, if that happens now, this is what's going to happen here. So let's now transit to this. Let's magnify this neutrophil into this so that we can fully understand. Okay, so now let's say this is a bacteria again. This is a bacteria. Okay, this is a bacteria. okay, which was initially outside. Now it will enter into what? Into the cytoplasm, because the vacuum, there was what pseudopodia that were extended like that, and the complete surround the what? That bacteria. Okay, this bacteria will enter into this vacuum. Okay, this bacteria will enter into what? This vacuum. What do we call that vacuum? Okay, we call it the phagocytic vacuum. Okay, phagocytic what? Vacuum. What we call it? Phagocytic vacuum. But we're going to use a certain term that they use. They use what? Phagosome. Okay, what do they use? They use what we call this term, which we call phagosome. Okay, so now just to recap, so the moment this bacteria enters there due to opsonization, then it enters into a vacuum. Basically, that's where what engulfment process what it ends from. The moment it just enters, after recognition, it has been recognized, it has undergone opsonization, then it enters into the cytoplasm. Then in the cytoplasm, it's not just on its own, it is inside the vacuum, which we call phagosome. So that's where engulfment enters. The moment it enters like that, that's where what engulfment ends from. Now we got what formation of what the phagocytic vacuum, which is this one. Okay, this is initially the vacuum that was formed initially. It coming from what extension of this cell membrane like that, where the bacteria inside where it is. Okay. Now what happens is really interesting, because inside this vacuum there is nothing there. Okay, in the phagosome there is nothing that can kill a bacteria. But our main goal of this is to kill this bacteria, which is here. Okay. Then what's going to happen is this. This phagosome is going to combine with something that is very, very important. What do we call this vacuum? We call it what? Lysosome. What do we call this vacuum? We call it what? Lysosome. Okay? Lysosome. Lysosome. Sorry. Lysosome. Okay? We call it a what? Lysosome. Now, this lysosome now, this vacuum which we are calling lysosome, they are going to start combining with what? A phagosome. There will be a combination like that, okay? There will be a combination between this vacuum and the other vacuum. This vacuum, it contains a lot of substances here which are very, very important here that are going to help to kill what this engulfed bacteria inside. So it contains a lot of stuff here. It contains enzymes like elastases. It contains reactive oxygen species. It contains what? A lot of stuff that we'll talk about. Okay, that's the reason why it's going to combine here. If it combines now with this one, it's going to form this larger vacuum. This one, if it combines like that, okay, what do we call that? If there is that combination now, if there's that combination, then what do we call this vacuum where you combine now the phagosome and the lysosome? Okay, what do we call that? We call it the phagolysosome. Okay, phagolysosome. What do we call this vacuum? What do we call this vacuum? We call it what? Phagolysosome. Okay, phagolysosome. It's just a combination of this phago and the lysosome. Okay, so that's a combination of that. So now here we've got now enzymes that we need, and we've got what like the bacteria that we need to carry. So yeah, so we'll put again this bacteria which is here in red. So this is the bacteria now which is inside, and remember that this is the lysosome type of vacuum. It contains everything. Okay, now let's dive into details. So what are the important substances that are in the what? Lysosome. I talked about reactive oxygen species. So remember, we talked about accumulation of free radicals. You need make sure that you watch that video so that you can fully understand about what? The free radicals. But just to recap, what are the free radicals? Free radicals are what? Are unstable compounds that are what? That helps to what? Eradicate or kill what? Foreign invaders. They are always unstable. They react. Okay. So now the question is, where do they come from? And remember that there are a lot of types of what? Free radicals. But the most important free radical is what? Oxygen reactive species. And what? The other one is what? Nitrogen oxygen what? Nitrogen what? Derived radicals. Okay. Okay, so now you've seen this cute enzyme which is just waiting for something to happen. Okay, this cute enzyme is what we call phagocyte oxidase. This cute enzyme. So this cute enzyme is called what? Phagocyte oxidase. Okay. Sometimes it is called NAPH oxidase, but let's just call it phagocyte oxidase. Okay, this is a cute enzyme. Okay, it's waiting for something. Okay, it's waiting for something. So this cute enzyme, the moment it is activated, initially, remember that here this is a lysosome. Okay, initially it was outside. The moment it turns its cytoplasmic component, then it goes into the lysosome there, then it's going to be activated. So initially, before activation, it was just outside here. Okay, but the moment like the fusion happens, it will change. It will go into the inside there in the lysosome. It will change its cytoplasmic component. Then in the moment it's like that, look at him. He's so sad and angry. Okay. Then the moment is activated, this one is going to do something. This one is going to convert something, which is this one is going to use NAPH, which is nicotinamide adenyl phosphate. Okay, nicotinamide NADPH. Okay, it's going to get that one, nicotinamide adenyl dinucleotide phosphate. Going to get that one, okay, and use this molecule to produce something which is very, very important. It's going to use that one to produce oxygen. Okay, this oxygen spontaneously is going to be converted into another species, which is this one, which is this one, with a free electron which is there. So what we call this, so NADPH is going to be used to produce oxygen, and this oxygen now is going to be spontaneously be converted into what we call superoxide. What we call it? We call it superoxide, type of free radical. Okay, type of free radical. So this is a free radical. Okay. This is a free radical. So that's what's going to happen there. Okay, what else is going to happen here? Okay, this superoxide now, although it's a free radical, it's not sufficient enough to kill this bacteria which is waiting there. Okay, so this neutrophil is so clever that, okay, since this is not so powerful, we're going to convert it into something that is more powerful and something that we can use it to kill a bacteria. Okay, so what's going to happen from there? So what's going to happen is, okay, this superoxide is going to be converted into something. Okay, it's going to be converted into hydrogen peroxide by using this enzyme which we call superoxide dismutase. So we call it superoxide dismutase. It's the one that's going to convert this superoxide into hydrogen peroxide. Okay, hydrogen peroxide, remember that is something that can move through from one biological membrane to another. Okay, but before it leaves the phagolysosome, it's going to be converted into something very, very important. Okay, so again, I know it's a lot, but this is as simple as a reaction that's going to happen. So as you can see, something is going to happen here. Okay, this hydrogen peroxide is going to be converted into something that is so powerful. Okay, if you remember, if you are talking about, this is a neutrophil. Okay, this is a neutrophil. This neutrophil, it has what we call eosinophilic type of granules. Eosinophilic type of granules. So in these granules here, so let's say these are eosinophilic granules. In these granules, there's an enzyme which is very important because it's going to make sure that this hydrogen peroxide is converted into what we call hypochloric it's going to be converted into hypochloric acid. Okay, it's going to be converted into what we call hypochloric acid. What we call that enzyme, which is found in the eosinophilic type of granules in the neutrophils. What we call that? We call it myroperoxidase. This is the enzyme which is very, very important. Where can you find it? We can find it in the eosinophilic granules. Okay, you can find it in the eosinophilic granules. So this myeloperoxidase is the one that's going to convert this hydrogen peroxide into what? Hypochloric acid. Okay. So if this is now the case, this hypochloric acid is so powerful that this bacteria that was here, okay, this foreign invader that was here, it can be what? It can be killed efficiently. Okay. Yeah. So that's the first mechanism. The other mechanism is this one. This, you've seen this hydrogen peroxide. Hydrogen peroxide can be converted into something as well. Can be converted into something which is the hydrogen what? I mean, that is the hydroxide what? Ion, which is under what we call under Fenton reaction by the use of what? Ion as what? Something that is a cofactor. Okay. So hydrogen oxide also is going to be what? I mean, hydroxide ion is also going to be what? Produced, so that it can also help and to start killing what? This bacteria as well. Okay. Another aspect is going to happen. You see, this sweet enzyme is also waiting for something. Okay. Neutrophil, something that is what? Something that is clever. Okay. What's going to happen here again? So you see this sweet enzyme, we call it what? INO. Okay. Inducible nitric oxide what? Oxidase. I mean, inducible nitric oxide what? Oxidase. Okay, that's what we call it. So INO. Okay, inducible nitric oxide what? Oxidase. This sweet enzyme. Okay, and this one, what it does is something else, because sometimes these hypochloric acid and also hydroxide ions, they are not powerful. Okay, again we're going to get what? Nitric oxide from what? The cytoplasm here. So we're going to get what? Nitric oxide from the cytoplasm here. We're going to get nitric oxide from the cytoplasm, then it will enter here. But basically it comes in form of what? Arginine, something like that. Okay, but it will enter, the end product is that it will enter into the what? Phagolysosome. The moment it enters into the phagolysosome, it's going to be converted into something so powerful. Okay, so this enzyme is going to convert this nitric oxide into something that is so powerful. What do we call that? Hypochloric what? I mean, hypochlorite what? Type of free radical. So what do we call that? So hypochlorite type of what? Free radical. So we are saying also hypochlorite type of free radical. So let's just put NO3, then the radical there. Okay. So this one is also used to kill what? This bacteria. So the whole process is to make sure that we kill that bacteria. Okay, all these processes. Okay, but here we are just talking about the free radicals, the mechanism in which a neutrophil white blood cell leukocyte, they kill foreign invaders by using what? Free radical mechanism. There are also other things that apart from free radicals that can be found here. Okay, apart from what? Apart from free radicals, what else can be found there? Okay, there are others that can be found there apart from free radicals. What else? So there are also enzymes that are there. Okay, that will help also to clear out what? To clear out this bacteria. So what are those important enzymes? So let's just put there's what we call catalase, but let's just say elastase, which is a very, very important one. Elastase. So elastase is a very important enzyme because the one that's going to kill also foreign invaders. Okay. Okay, so and if you talk about eosinophils, okay, if you talk about eosinophils, they use a certain protein, a basophilic type of protein that's going to kill what? Parasitic infection. Also, they are here. There are a lot of what substances that are within what? Within the phagocytic, I mean, phagolysosome. Okay, a lot of them. Okay, so yeah, so that's where formation of so we are here. So let's try to connect whatever we're talking about. Formation of phagocytic vacuum, we've talked about it, and the last one we've talked about how they kill using what? Free radicals. Using enzymes, that's simple. The enzymes that are within there, they will go direct to the what? To the bacteria and kill it. Okay. Yeah. So now, what is the last thing that we need to talk about? The last thing that we need to talk about, I mean the second last that we need to talk about is what? How neutrophils kill by using what? Traps. They kill bacteria or foreign invaders by using what we call traps. Okay. So neutrophil extracellular what? Traps, which are also called what? Nets. So how do they do this? Okay, how do they do this? So here we're done with phagocytosis. Let's go to what? Let's go to this one. So now, how do they kill? By production of nets. So remember that if this is a neutrophil, okay, so we're just giving an example. If this is a neutrophil, okay, if this is a neutrophil, like that, that's a neutrophil, and this is a bacteria, which is what? Outside, like that, okay? Phagocytosis is not going to happen there, okay? So what they do is this. They are going to sacrifice themselves. They are going to kill themselves, okay? The moment this kills itself, then it's going to produce something which we are calling traps, okay? Traps. So what are those traps? We call them what? We call them extracellular what? Extracellular fibrilla, okay? Or fibrilla. So extracellular what? Fibrilla type of what? Network. So these are the things that they are going to produce. So they look like this, okay? They look like this. These traps, they look like that, okay? So they are going to produce that by sacrificing themselves. So these traps, you need to understand what can be found inside these traps. So you need to understand them. So inside these traps, you can find what? A lot of what? You can find a lot of what? Enzymes. You can find a lot of enzymes in these traps, okay? And also you can also find what we call like chromatins. You can also find what we call chromatin. Basically, you can find DNA there, okay? You can find DNA, you can find histones, okay? A lot of stuff. So you can find them like that, okay? So now, why do they produce traps? So this bacteria, as you can see, this bacteria is able to move from one place to another. If there are some traps, this bacteria, if let's say it meets these traps, so what's going to happen? This bacteria will be trapped there. And if the moment it's trapped, then it's going to be killed by the enzymes within the what? The traps, okay? It's going to be killed because these enzymes, remember the most important enzyme that we talked about was what? Elastase, okay? The most important enzyme that is there is what? Elastase. It's going to be found there, and it's the one that's going to kill what? That, okay? Sometimes these traps, maybe if the elastase are not what? Enough, then also other white blood cells that are nearby these traps, they are going to start picking up those what? Those bacteria, so that they can initiate the process of what? Phagocytosis. So these traps are very, very important. And the other important aspect about net is that it has been shown that these traps, if someone is having sepsis, okay, those disseminated type of infection, okay, they are having sepsis, it has been found that these traps, they are going to be found within blood, okay, and it's going to help you in the diagnosis of what? Sepsis. And also it has some relationship with what? Lupus, okay, lupus erythematosus, okay, lupus. Let's say it's systemic lupus erythematosus. Let's just say SLE. Systemic lupus erythematosus. They found out to say these nets or neutrophil extracellular traps, they can also be found in someone having what? Lupus, which is systemic lupus erythematosus. Okay, very important concept. What is the last thing that we need to talk about? We said production of what? Chemical mediators. So what is the last thing? Chemical mediators. Chemical mediators. Production of what? Chemical mediators. Very important what? Concept. Now, if we talk about the chemical mediators, so chemical mediators, we'll talk about them in details in the what? Next video. But here, let's just summarize it. So, after everything here happens, these neutrophils or these leukocytes, they are also able to produce what? They are also able to produce these chemical mediators. Why producing chemical mediators? The reason why they produce chemical mediators is because they need to call more what? More leukocytes to the site of what? Infection. Okay? Very important one. So now, the question is, what are the types of chemical mediators that we know? Okay? The types of chemical mediators that we know is that there's what we call cellular type of chemical mediators, or cell-derived. So let's say cell-derived mediators. So mean that the mediators that will come from the cells like leukotrienes, histamines, serotonin, those they come from what? Cell. We call them cell-derived mediators, okay? Then what else? There's also what we call plasma protein derived. So we are saying the other one is what? Plasma protein derived type of what? Chemical mediators. Okay. So like what? The complement, the kinin, we'll talk about them one. So the kinin, the proteins, blood kinin, all those things, they come from what? Plasma. The C3 that we talked about, that they come from what? Plasma. They're under what? They're under plasma protein derived type of what? Mediators. We'll talk about that them in the next what? Video. So this is the end of the video. Stay tuned to the what? To the next next video that we're going to have, okay? And make sure that you study well.